Hello, this is talk number 13 in the series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we will be discussing enzyme kinetics and the mathematical formulations that derive from the study of enzyme kinetics. As stated earlier, enzymes are catalysts in biological systems. They lower the activation energy and Gibbs energy to affect the rate of and completion of reactions that occur in biological systems. There is a way to quantify all this, and we are discussing enzyme kinetics to achieve that particular goal. So enzyme kinetics, we talk about enzymes. They are proteins, and proteins are affected by their surrounding conditions. So the enzyme behavior, whether it works effectively or not, is subjected to different physiologic conditions. The most critical of these is the substrate concentration, how much substrate is around. And when we look at substrate, we have, as I showed earlier, what's called the velocity, which is the number of molecules that are converted in a second, and there's a rate constant. So at the very start of a reaction, we talk about V naught, which is the initial velocity, and then V max is the velocity of the reaction at the maximum. In biological systems, for quantitation and characterization of enzymes, oftentimes we will saturate the system with substrate, and Vmax is an extrapolated number. So when we look at the amount of substrate present, and we have an infinite amount of substrate, we get the initial velocity V0, V naught, approaching Vmax. This line, Vmax is here, and the line eventually will approach this number. So the Vmax is at this point where there's an inflection point is 1/2 of that, and there's a certain the Michaelis constant Km is what characterizes the case where the initial velocity is defined as 1/2 Vmax. So this number is right at this point. If you had an infinite amount of velocity of transference without any kind of limitation, we could extrapolate that V naught is equivalent to Vmax times the substrate concentration over Km. At the case where V naught equals Vmax, we can get Km equal to S at this point. But for the purposes of this talk, we can visualize an enzyme being able to function as long as the enzyme has enough copies present. It can handle however many molecules of the substrate need to be transformed up to the limitation that's provided by the amount of enzyme and the velocity that the enzyme produces. So Km is the Michaelis constant, and we talked about intermediate energy transition forms, and we look at the combination of the enzyme and the substrate, which is a transient form, and this can be characterized by covalent bonds or any degree of multifactorial interaction between the enzyme and the substrate. So we talk about the first order reaction going from the reactant substrate and the enzyme to the intermediate complex, and then returning in the second reaction to this point where you have a product and the enzyme. The second reaction in this series is termed the rate limiting step. And as the substrate gets depleted, you get more and more dependence on this number. So you eliminate this component, and you're looking at the dissociation of the enzyme substrate complex to this. And as shown earlier, when you look at the the math of this, the V naught is equivalent to the maximum velocity times the product of the substrate divided by the summation of the Michaelis constant and the substrate here. When we look at this this graph here, it's a double reciprocal plot. This is called the Lineweaver-Burk plot. The Lineweaver-Burk plot is the plot that you get when you invert the velocity. This is a cleaner, mathematically more recognizable way of dealing with the mathematics. Instead of dealing with an asymptotic line that I'd shown you in the earlier graph, you're looking simply at an inverse where this is this axis, the asymptote has the inverse of the substrate concentration, and this is in millimoles. And at the point where this intersects here, it's the equivalent to 1 over the Michaelis constant in a negative integer here. This the ordinate here is going to be the 1 over V naught, which is in micromoles per minute. So as I said in the last talk, this is the standard unit for this, for velocity. It's micromoles per minute that are transformed, and this is a quantifiable entity. If you look at reactions, you can stop reactions early in the in vitro phase and do a quantitation of your material at that point. You can get a very good handle on what your velocity is. This slope, therefore, is the Michaelis constant over V max. So by using by looking at the concentration at various points of the substrate along this line here, you can get a linear curve with the intercept right here at 1 over initial velocity, and right here at the abscissa for 1 over the Michaelis constant. This is the Lineweaver-Burk plot. So when you look at the Michaelis constant concerns the different orders. So in the first order, we have the breakdown of the relation between the substrate and enzyme with the transition point here. So K minus 1 references this reverse reaction, and K1 is the forward reaction of this. So K1 goes to the forward point, minus 1 to the negative point. And then you also have K2, which is the breakdown of the intermediate to the free enzyme plus the product. So when we look at the Michaelis constant, we're looking at this forward reaction here and the constant of the forward reaction here. These two are summed, and then you divide by the constant of the forward reaction of the first order of this. So this is the first order, this is the second order, and the Michaelis constant is the ratio of both of these over the first order constant. And again, the Michaelis-Menten equation provides the data as a double reciprocal plot. So going back to the Lineweaver-Burk equation, we have this relation here, where if you took the initial equation of the initial velocity is equivalent to the Vmax times substrate divided by Km, and you inverted these terms, you could get the construct of the Lineweaver-Burk equation, which is placed in graphical form in the first graph that I showed you, where these are all inverted, and you have the intercepts at Km and Vmax in reciprocal form. For the rate limiting step, the K2 has to be much less than K1 or K minus 1 by definition. So minus 1 was the breakdown of the enzyme substrate complex going in reverse form to the substrate and enzyme, not the forward reaction, but the reverse reaction. But that number has to be much less than that has to be much greater than the forward reaction of the second order equation, which is the enzyme substrate complex going forward to product and enzyme. So the rate limiting step is this forward reaction of the second order, which is AES breaking down to product and free enzyme. So when you have the K2 being vanishingly small, then the Km reduces simply to the ratio of the reverse constant, which is the electron, the ES complex going back to substrate and enzyme over the forward, or the forward of the initial reaction, which is substrate and enzyme going forward. So this is the reverse of the first order. This is the forward reaction of first order, and that is simply what Michaelis constant reduces to if this number is exceedingly small and can effectively be ignored for further consideration. The dissociation constant is also a situation where you get Km equals Kd, and this is the breakdown of the enzyme substrate complex, either it going back to the reactant or going forward to the product. The turnover number is what happens when you have saturated substrate. If you simply flood the in vitro system with substrate, and then this is the maximum number where the substrate molecules are reacting per unit time. And under that circumstance, you have maximum velocity, and K3 is a term times the enzyme, where the enzyme becomes, the amount of enzyme present becomes the limiting step because you flooded this with substrate. So you basically have the enzyme product complex breaking down into enzyme plus free product. And this is the enzyme in product form here. So this comes into play with the maximum velocity when you simply flooded the substrate molecule, and when you do that, this number becomes exceptionally large, and you reduce this to Vmax is equal to VW, and this number gets exceedingly large, and then the reaction is pretty much dependent on the amount of enzyme that is limiting this step. If you have two substrates, which is usually what's occurring in these biological systems, 2/3 of these enzymes that we study have two substrates and two products. We'll be reviewing many of those. These are generally redox reactions. We'll be talking about these redox reactions, which are not usually occurring at the reaction site in particular, but oftentimes adjacent to that. So we'll be walking through that. But you have one substrate oxidized, the other substrate is reduced. And you can get ternary complexes where substrates bind concurrently, and double displacement where the substrates bind sequentially. And when you do a Lineweaver Burk plot of those, this is a ternary complex. So instead of one Michaelis constant here, the Michaelis constant changes with increased amount of substrate of the second substrate, and this is the first substrate here. So at a given first substrate, the amount of increased substrate of the second one drops this, and also will increase the Michaelis constant reciprocal over here, and the initial velocity will also change. This initial velocity will increase as the second substrate. This is a ternary, where the substrates react sequentially, and then you have the double displacement over here. And the double displacement is a ping pong line where one substrate reacts, and then you get it acted on, and then the next substrate comes in afterwards. And these are interesting developments. The slopes or equivalents of these lines run in parallel as the number 2 substrate is increased. We talk about pH effect on the enzymes. They all have optimum enzymes. So pepsin, which is a secretion in the gastric fluid, acts optimally at pH of a little bit under 2. There's special linings that prohibit the stomach being chewed through, which is why ulcers are problematic. whereas a glycolysis enzyme such as glucose 6 phosphatase has an optimum pH of close to 8. For pre-steady state kinetics, we begin a reaction. Again, this is how we do this in the lab. We begin a reaction, stop rapidly, measure constituents, and then we can estimate the enzyme substrate intermediate transition state, rate limiting steps. So you can measure everything and then apply this to the Michaelis-Menten and Lineweaver-Burk plots specifically, and be able to characterize an enzyme. You can also get reversible inhibition, and inhibition, there are different types, and they all have biological appearances on the Lineweaver-Burk and can be characterized quite nicely. So the most common of these is competitive inhibition, where the inhibitor directly competes with substrate for the binding site. The enzyme is not inactivated, and the Km increases, Vmax is constant, and you can override the inhibitor by adding more substrate. So, for example, if you have methanol poisoning, which is very serious, you can give a competing substrate, which is ethanol, and that would be a treatment modality you could use for that. So in these cases, these molecules, the competing substrate it has structural is a structural homologue to that of the inhibiting compound. Another way is uncompetitive inhibition, where you see these in two substrate reactions. The inhibitor binds to a site separate from that of the substrate. It will bind to the enzyme substrate complex. The inhibitor removes the enzyme from the reaction, whereas competitive inhibition removes the substrate. Both Vmax and Km, Michaelis constant, decrease with uncompetitive. So in this case, the inhibitor binds away from the binding site of the substrate and removes the enzyme from reaction. Mixed inhibition is a case where the inhibitor binds the enzyme site separate from that of the substrate, but it binds either free enzyme or the enzyme substrate complex. Vmax and Km vary under that circumstance. So when you look at competitive inhibition, the Vmax is the same in all cases. As the inhibitor concentration increases, you get change in the Michaelis constant over here. This is a case where you are overriding. This is, again, a substrate structural analog, and you get overriding of this inhibition if you increase the amount of substrate. And when you do that, you still get the same Vmax, but the Km is what's changing here. So this is a classic graph that you get with competitive inhibition when you're tracking the velocity in reciprocal versus the substrate to reciprocal that's on the ordinate, or the abscissa rather. For uncompetitive inhibition, both of these numbers change. The Vmax changes, the Michaelis constant changes, as inhibition increases, you get a series of parallel lines. So the slopes change, and this is a case where the enzyme is taken out of commission by the inhibitor, which is binding, again, away from the substrate's binding site. So increasing the amount of substrate, you get changes of maximum velocity and Michaelis constant. When you get changes of all these with an interception point and lack of a parallel array, then you have a different situation, and this is a situation where you get mixed inhibition. So you have inhibition where the enzyme is being affected, the enzyme substrate complex is being affected, and again, this is a track of increased inhibition. As inhibition concentration increases, the velocity of the reaction decreases, and this number increases with the increased amount of inhibitor. For irreversible inhibition, these are covalent links. The mechanism, you have you have inactivators that participate in the early reaction, and they form a compound stop the reaction. This is a covalent bonding situation where you've got analogs to the transition state structure, and it mimics the transition states structure. So these are irreversible inhibitions that occur. And then we also have enzymatic reaction examples. So some of the reactions that we referred to, one example is chymotrypsin. That's a pancreatic secretion substance, and it cleaves peptides, and you get acetylation of the serine residue. Hexokinase is an induced fit by glucose with a phosphoryl transfer. And you have allosteric enzymes, which have inhibitory or stimulatory conformational changes with binding. One example of an allosteric protein, albeit not an enzyme, would be hemoglobin, which changes its conformation with increased oxygen binding. So the allosteric enzymes do change the enzyme structure as you have substances bind to it, and this facilitates its biological activity. So when you look at allosteric enzymes, they don't adhere to Michaelis-Menten kinetics at all. You get a sigmoid curve instead of a hyperbolic graph. So we looked at, for example, these are examples that we showed earlier, where with the increased amount of substrate, the amount of velocity approaches Vmax. But with allosteric enzymes, you get more of a sigmoid curve where it'll go up like this. It'll go that way and then up, and then like this. Instead of a hyperbolic curve, it will actually have a delay and then an increase, and then the slope lowers, and then you get out here. This is, again, the kind of track you would get if you were binding oxygen to hemoglobin, and this is an element of cooperativity. It would increase. It's a slight sigmoid curve with a little bit of delay, and then you get the first oxygen, second oxygen, then when you get past that, you have a curve that resembles that of a first order enzyme. So when we look at those type of allosteric enzymes, those conformational changes render classic Michaelis-Menten mechanics inoperable. We also have enzyme modifications here, and we talk about what occurs with enzymes after they are produced as proteins. So you can attach covalent bonding by modifying groups such as acetyl, acetyl, ADP, carboxyl groups, methyl groups. These are post-translational modifications of a protein, and the enzymes are just not pure amino acids, but they can also have other constituent groups on them, as referenced here. The most common type of regulatory modification that occurs is phosphorylation, and there are protein kinases, and there are a whole variety of protein kinases which exist. And those protein kinases will phosphorylate enzymes. You can transfer them to the amino acid hydroxyl groups that are present on there and form esters. You get this bulky charge moiety with energy transfer potential. So the hydrolysis of that anhydride phosphoester will create energy, and that will help drive a lot of these reactions to completion. As mentioned, these are always energy-consuming reactions, and that is often done for cases where the energy cannot occur spontaneously. You have to drive it with ATP, for example. These phosphorylated proteins are greatly involved in regulation of metabolic pathways. We'll see a lot of that. Zymogens or precursor enzymes, those are cleaved to form an active enzyme. So, for example, you get trypsinogen, which is formed in the pancreas, and there's a cleavage of that to the active form trypsin. Chymotrypsinogen is the zymogen precursor, and you get chymotrypsin from that. And then finally, coagulation is an example where you get many zymogens in coagulation cascade. These are multi-layered, multifactorial cascade where the initial reaction of one component causes a numerical cascade. So by the end of the process, you get many thousands of transitions occurring based off of the first reaction in the cascade of separate enzyme reactions. So the initial cleavage of proteins in the coagulation cascade causes multiples of cleavage in subsequent proteins, and this cascade is what helps formulate blood clotting in the biological system. There are two pathways involved. There's an intrinsic pathway that's modulated by thrombin, and the extrinsic pathway is modulated by vascular injury, which allows for the presence of factors that help accelerate, initiate, and exacerbate the coagulation cascade. So there are two pathways at work. The activators and inhibitors involved, they can culminate in fibrinogen converted cross-linked fibrin polymer, and these will lock in and create a clot. And there are serine proteases in this cascade. They include factors 7, 9, 10, and 11. So when we look at the actual cascade, we have the intrinsic pathway, which has thrombin. You have the extrinsic pathway of vascular injury, and you have factor 11 going to activated factor 11A. Antithrombin 3 inhibits the next group of this reaction. 9 going to 9A with blockage by again antithrombin 3. You have the 7A, which is an activated form from the vascular injury that goes over here. These are all very complicated. Suffice to say is that every one of these causes multiples. With calcium cofactor, you could get transformation. So you have the extrinsic and intrinsic pathway here, and these are branch, interact, and you have a common pathway that starts at this point, and you get prothrombin going to thrombin. Thrombin activates factor 13, also activates, transfers fibrinogen, serine protease of fibrin monomer, fibrin polymer, and then the cross-link. So the end result is a clotting occurring here, and you get activation of these entities, thrombomodulin, and you have protein C, protein S. So people with clotting problems will oftentimes have problems with factor 8, for example. They can have decreases or deficiencies of protein C, protein S, or they can have linkage problems. So a series of, this is a very delicate, complex process, and we have quantitative assessment to determine which of these cascades you utilize in partial thromboplastin time or prothrombin time, and then clotting is a broader way of determining efficacy of the process that occurs at the very end. So that is it. We will be moving on to carbohydrates next. Thank you very much.